And we'll be on our way. Okay. Again, this meetup is about real-world data and its application in the pharmaceutical industry. It is one of a series of meetups that NISS is co-sponsoring with Merck on emerging topics in the pharma industry. As I mentioned earlier, it's being co-sponsored by the Health Policy and Statistics section of ASA. Randomized controlled trials have long been the gold standard, providing evidence of safety and efficacy for new drugs, vaccines, and biologics. But complete reliance on controlled randomized trials comes at a price. They're costly, and they're limited in the very specific clinical circumstances that get studied. Real-world data, on the other hand, comes from a much more heterogeneous population, often in an observational manner, but there is a lot of it, and although it doesn't have the same assurances against biases as randomized clinical trials, it certainly can supply a rich and diverse set of information that may be useful in terms of discovering, developing, and post-marketing of compounds. So today's meetup is to focus on that particular topic. We have a great set of panelists, two great speakers, and a great discussant to walk us through that. I'll mention that all of these meetups are recorded, and the recordings and the slides are available at www.niss.org. And I'll also mention just that, this is a really hot topic in the pharma industry, and one result of that is just this week there was a great conference at the Duke Margolis Center for Health Policy, and I put the link for that on for those who might be interested later to look that up. They have a very interesting white paper and a lot of discussion around that. So with that, let me just mention again, it's co-sponsored by NISS. NISS stands for the National Institute of Statistical Sciences. Merck interfaces with NISS mostly through the affiliates program. So as members of pharmaceutical companies, you should think about whether you want to become affiliates too. It has a lot of different benefits, but one of those benefits is getting special dispensation sometimes to go to some of their workshops. A couple of their workshops coming up, one next month is on dynamic social networks, and you can see that that's at Ohio State. And then next spring, they're having a workshop on biostatistics and bioinformatics in Atlanta, Georgia. If you want more information on these, go to the NISS website. You can see the address there. And we want to again thank NISS for sponsoring this meetup, and that is a part of their central mission. To introduce the next section, or to tell us about the health policy statistics section, I'd like to introduce Kelly Zhou. She, as mentioned earlier, is a senior director and methods and algorithms group lead, real world data and analytics at Pfizer. She's an accredited professional statistician and an ASA fellow. She chairs the health policy statistics section and the statistical partnership among academia, industry, and government, sometimes called SPEG committee. And she's also a member of the Methods and Data Council, the Academy of Health. So with that, Kelly, can you tell us a little bit about HPSS?
Yes, thank you, Dan, and thanks to both NISS and Merck for co-sponsoring, hosting today's workshop. And we're also very glad that HPSS, which stands for Health Policy Stats section of the ASA, is co-sponsoring today's workshop. And this is a slide that talks about the HPSS, which has about 700 members out of a total of 19,000 ASA members. So we have various initiatives, including sponsoring sessions, workshops. Our key conference that's held every 2 or 3 years is the International Conference in Health Policy Statistics. That's going to be held in 2020 next, and it was also held earlier this year. I'm very glad to also remind that Anna Bond, who's one of of the presenters today is, in fact, our 2018 Mid-Career Award winner. So congratulations to Anabar. In addition, we provide other types of networking, training, career development opportunities, and also establishing joint fellowships with the ASA as the organizational affiliate to the organization called Academy Health. And there is, in fact, the Real World Evidence Fellowship. We also encourage quality and impactful publications on various topics, including the one that's going to be, discussed today by Anabar that has generated so much interest in the public and also in media, as well as in the scientific community. And finally, we always try to find the ways to leverage data and tools through close collaborations and partnerships. So if you have any ideas or any interest in collaborating with NISS as well as HPSS, please do let us know. So with that, I'm very glad to lead you to today's presentations by Anabar and Doug. Thank you.
Thank you, Kelly. So again, we have a great set of speakers today. We'll start off with Anirban Basu talking about real-world evidence and regulatory infrastructure, and then Doug Ferries will talk about real-world evidence in pharma, best practices and innovation, and we'll follow that with some comments from Kelly and questions and discussion. So with that, let me introduce Anirban Basu. Dr. Basu is a health economist and a statistician at the University of Washington, who specializes in research on comparative and cost-effectiveness analyses, causal inference methods, program evaluation, and outcomes research. He directs the Comparative Health Outcomes Policy and Economics, sometimes called CHOICE Institute, at the University of Washington. He has appointments in the departments of pharmacy, health services, and economics. He's a faculty research fellow at the National Bureau of Economic Research and also an ASA fellow. As Kelly mentioned, he won the 2018 HBSS Mid-Year Career Award for healthcare policy and health services research. And he received his PhD in public policy from the University of Chicago and an MS in biostatistics from the University of North Carolina at Chapel Hill. So with that, let me pass the ball to Dr. Basu, and he can tell us about real-world evidence and disrupting the outdated regulatory structure.
Well, thanks, Dan. Thank you for the opportunity to present today. We're appreciative of NISS and Merck and HPSS to bring together this forum. What I'm going to talk today is really not about what's going on today in real-world evidence, but what are the challenges that we face and how we could potentially address those challenges going forward. And especially, I'm going to talk about a normative approach of disrupting the current regulatory infrastructure that could really potentially provide us with information that's decision relevant. Let me see. Oh, yeah. So as you know, as a new technology comes to the market, and especially, for example, a drug, there are several questions that decision makers face. And these decision makers are different decision makers. For example, one can think how a manufacturer should price their drug at launch. How health plans should whether the health plan should put a drug on their formularies, which tiering they should do. And from the clinical decision-making point of view, which patient would benefit most from this new drug? So these questions are all real-world questions. And the challenge that we have in answering is that we need to produce evidence to be answering all these questions, but in a timely fashion. And in doing so, we have to somehow maintain the quality of the evidence so that relying on the evidence could be reliable. And in addition, when we make these decisions, they have to be equitable across a vast population and a diverse set of patients. So the question is, how do we achieve all of this while answering these real-world questions? obviously, I'm sure all of you know that the current infrastructure of especially drugs and regulatory infrastructure for drugs is the phase-based approach to information or evidence production. The phase 1 usually are animal trials, then phase 2 becomes the safety trials, and then after completion of phase 2, and there are sometimes some efficacy endpoints are added to phase 2, and then the main efficacy trials are the phase 3, and at the end of the phase 3 trials, the manufacturers submits an NDA application with FDA, and that gets reviewed, and then approval determination is made. Now, obviously, as Dan mentioned, there are a lot of challenges to these infrastructure. First of all, let me see if I can. Yeah, so the evidence collected, the evidence produced in these trials are typically based on about 1% of the target population. And even so, when these trials are conducted, the patients in the population have inequitable access to phase 3 trials. These trials are often conducted in academic setting. Although there are multi-center trials, there is a huge NIH portfolio research now talking about how there are disparities of access to these new innovations through phase 3 trials across different parts of the country and for different patients. Also, treatment responses are believed to be heterogeneous across patients, and often these phase 3 trials are not powered or not big enough to really look at heterogeneity that could guide clinical decision making. And then there is always this debate about why efficacy and not effectiveness should guide decision making in the real world. So the challenge that we face in designing this trial, and this infrastructure of kind of hypothesis-driven evidence production has been there for the last 50 and more years, is that the whole statistical theory of hypothesis testing is not really a theory of decision making. And what we face is that we achieve precision in answering a narrow question in phase 3 trials. But that always, when we rely on that evidence to answer all these big real-world questions, that always leads to substantial imprecision in answering this more decision-relevant real-world question. So in this context, in comes this whole idea about real-world evidence, and especially with the impetus that 21st century cures provides, that we need to really understand what's really happening in the real world and help kind of inform these different real-world questions. So the whole reason we need real-world evidence is to decrease imprecision or decision uncertainty in answering these different decision-relevant questions. In fact, our current FDA commissioner did mention that, should a product be marketed based on data that speaks to a limited and rigidly constructed circumstance, when the clinical use and in turn the evidence we might have to evaluate the product could have been much far richer, far more diverse, and more informative. So this kind of sets the stage for using real-world evidence. Unfortunately, till date, the progress of actually incorporating real-world evidence and producing it is quite limited. We use some observational studies to design future phase 3 trials, and sometimes people have been talking about using historical real-world controls for orphan drugs. Most of the real-world evidence production has been relegated post-marketing. Obviously, you have to market the drug or put it out in the real world in order to produce real-world evidence. And then, as a result, when you put it out in the real world, the real-world evidence on comparative effectiveness and safety always have these problems of omitted variable bias and selection bias, and I think our Douglas is going to talk a lot about that. But the real use of reliable real-world evidence in decision-making has been limited because I think we have not thought about how to produce real-world evidence in the right way. We have been reactive and say, well, there is real-world evidence, and how do I kind of fit that evidence that's produced under the current infrastructure into our decision-making purposes? instead of thinking, how can we actually produce real-world evidence in a different way. So one of the things as a thought experiment that I wrote last year was this idea of thinking anew about of our regulatory infrastructure, and in particular I proposed this OIR infrastructure, only in research infrastructure, where a company can go through phase 1 and possibly phase 2, where they generate safety signals. And these phase 2 could be actually bolstered to put some efficacy signals in there, or have a more broader safety portfolio. But then at the end of phase 2, the manufacturers can submit a safety-based NDA application. And once that's approved, the most disruptive part of this infrastructure is the FDA giving access to this drug in the real world with the OIR nomenclature designation, but in a randomized fashion. And I'm going to talk a lot about how we can achieve that and what are the challenges of doing that. But suppose for a second that is achievable, that they can provide this OIR designation and have a randomized market access. After that, the manufacturer can actually conduct open-label prospective observational studies to study the effect of this drug on outcomes, but rely on the randomization already built into the system by FDA to really tease out causal signals from the data. And then they submit a final NDA application with real-world evidence and determine approved determination. In fact, the timeline for this process should not be very different than the timeline of the current phase 1, phase 2, and phase 3 trials. But there are so many other advantages that one could think of in terms of the data that such an infrastructure can produce. The most important and disruptive part of the infrastructure is obviously the random access to a random subset of the population. And perhaps the first criticism of this infrastructure is the ethicality behind it. How can we give non-uniform access to the whole population as an OIR product? But to understand the ethicality, we have to consider how that compares to what we currently do in terms of our patients or access to patients. In the current infrastructure, after phase 2, in the whole population of the patients, a selected group of patients are solicited and enrolled in the trial, and then a flip of a coin decides whether they get access to the drug or not. As I said, this selection is not always equitable. The patients are again may not be representative of the target population, and the access to the drug is limited to those few people who are enrolled in the phase 3 trials. Under the OIR infrastructure, under the same information on safety, a lot more people get access to that new drug initially, and in an equitable way, that because everybody has access in a randomized fashion. It maintains physician autonomy, and the most important thing, and it's line with real-world evidence, that the comparison of these new drugs is made to actual standard of practices across different regions and sectors. So let me talk a little bit about all the advantages such an infrastructure can produce. Obviously, I talked about early and equitable access to patients. But a randomized access at the population level has a lot of value. First of all, it directly answers the questions of the value of access. We have gone back and forth about models such as ICER or doing cost effectiveness analysis using FDA phase 3 trial data when the data is really sparse and we really do not have any effect on cost and effectiveness. And yet in this framework, at the time of launch, there is real-world evidence on the value of access to this drug because we had studied this in a randomized fashion. Obviously, we can expect large sample sizes to explore heterogeneity that allows providers, payers, and policymakers to better target use of new and expensive drugs. The randomization itself solves a lot of the angst of dealing with selection biases in real-world evidence. And more importantly, beyond what manufacturers submit to FDA in terms of their trials, prospective observational study trials, anybody, anywhere can actually replicate effectiveness studies in the OIR phase by relying on this population-level randomization. And I won't have time to go in details, but at this point, the OIR phase, the manufacturers are bound to price their product modestly during the OIR phase so that they ensure differential uptake between the two randomized groups, but yet enough people do uptake it so that a study could be conducted. So overall, this gives us tremendous advantage over the current infrastructure to really incorporate decision-relevant real-world evidence or produce decision-relevant real-world evidence. Now, there are, however, challenges to FDA. Obviously, they have to think, how do we randomize access? Do we allow certain physicians to prescribe the drug to a random group of patients? How do we choose a random group of patients? Whether it's through certain birth dates, asking physicians to follow instructions may always be very difficult. The other way to do it is to actually allow insurance companies to cover the drug only for a random group of patients, and that's maybe more palatable and legally enforceable in terms of doing randomized access. But if such infrastructure do happen, or do kind of carry out, this population, now the challenge for the FDA is that once you have this population-level randomization instrument, you can use to not only say approval decisions, but there's a whole host of evidence that could be produced for decision-making at different levels, which are not present in the current process. So I think in summary, what I would say is that the current infrastructure of phase 3 trials just fundamentally fails to remove decision uncertainty for most real-world decision-relevant questions. The evidence production is inequitable and slow, and we really need to disrupt this process to really generate evidence that can be acted on by different levels of real-world decision makers in a timely fashion. The OIR is a promising alternative for these purposes. In technologies outside health and medical care, OIR kind of infrastructure has been used. Like if you think about technology, people have done A/B testing all the time, and those A/B testing are not done in a very controlled environment. They are done in a population level. We have seen Oregon experiments where Medicaid was, there was a lottery to expand Medicaid and understand what's the value of insurances. So these kind of social experimentation has been done in other sectors. The question is, do we have the appetite or a way to do something related to that in this context? And if we can do that, I think we totally disrupt the way evidence is produced in this country. And it's not necessarily in this country. I mean, any infrastructure, any country that can envision this type of infrastructure can really catapult their evidence framework into the 21st century. For further details, you could read my Health Affairs blog. This is where I detail all the stuff, and I think I'm ready to discuss this further and ask questions.
Great. Thank you so much, Dr. Basu. Obviously, a very interesting and provocative idea, and hopefully one that we can have a lot of great conversation around. Our next speaker is Doug Ferries. Doug is got a PhD in statistics from Oklahoma State University. He's worked as a statistician in the pharmaceutical industry for over 28 years, currently a research fellow for real-world analytics at Eli Lilly, In this role, Doug is involved with the design and analysis of observational research, including comparative effectiveness analyses and the development of real-world analytical capabilities. His research interests are in statistical methodology for real-world data, including causal inference and unmeasured confounding. He has authored or co-authored over 100 peer-reviewed manuscripts and books, is active in the statistical community as a publication reviewer, speaker, workshop organizer, and teaches short courses in causal inference at national meetings. So we are very privileged to have Doug with us. I'm going to pass over the ball to him, and he's going to talk to us about real-world evidence in pharma, best practices, and innovations.
Thanks, Dan. I appreciate the opportunity to be here and talk about this from the pharma side and the innovations I think that we can have in this space. Oops, I may restart and try to do this correctly. There we go. So here's my outline. Again, I'm going to very briefly just discuss how we're using RWE at a pharmaceutical company. Really didn't want to turn and shift and spend most of the time on comparative effectiveness, looking at how we're doing, the growing regulatory interest in this, and then shift to a little bit of the innovation where I think we can make a difference going forward, less on the design side like Anirban was talking, but more on the analytics side. So again, given my math background, I like definitions that include equations. So this is my definition of real-world evidence that we use here. And again, the focus here is going to be on the design and the analytics piece, but just as a reminder, you know, the quality of the data and the right research questions are key to any piece of good research. Over the drug development process and over the last 10, 15 years, I would say, we've been very active in utilizing real-world data and real-world evidence. This starts with basic epidemiology studies, understanding the standard of care, treatment pattern, costs in any particular disease state that we're going to enter into. This has evolved also into support of the RCT phase, understanding the competition that we're going up against, what we expect, as well as the support for RCT planning and recruitment using real-world data. Submission work to date has been mostly on the HTA side, supporting our budget impact, cost effectiveness type of modeling, but also, in some cases, utilizing real-world data as control arms in rare diseases. Launching commercialization has been very active. Studying the comparative effectiveness of our medications, the cost effectiveness of our medications, getting into precision medicine, safety monitoring we've been doing for years, commercialization through FDAMA 114 in the U.S., and also lately supporting value-based contract agreements that have arisen. And of course, the current interest is, hey, can we do more than that on the regulatory side in terms of things such as label updates? So I'm going to shift now and talk about causal analysis mostly. So not using RWE to summarize the epi and treatment patterns and things like that, but really to make comparative and decision-making inferences off of real-world data. So this is just a cartoon I like. So there's been a lot of guidances over the years, starting about 10 to 15 years ago with the trend and strobe statements. So these are basic checklists about how to do quality observational research. And over the years, those have been updated by groups such as ISPOR and ISPI, getting into just general flow charts, such as saying, yes, you need to adjust for bias, you need to pre-specify your hypothesis, to the latest, the 2017 version, where the focus nowadays is really on transparency of the research and being able to document and pre-specify everything and put it out in the public to ensure the credibility of this research. So in addition to the guidance documents, we of course have the regulatory involvement now, which is growing. So at the top you see we do have a guidance document out there for the medical device division at the FDA. A lot of the focus of that document is about the quality of the data, which is a huge issue. And obviously with the 21st Century Cures Act, we will soon have more guidance coming from the FDA regarding that. But clearly there's plenty, like the Duke-Margolis conference and white paper, plenty of public places where regulatory has opened in discussions with industry and other groups about use of real-world evidence. So this is not things that are hidden, but are growing in public debates, which has been very, very, very helpful. So my first question on this is, how are we doing? And about 5 years ago, we saw the results of the OMOP work. And again, this was simulation-based and largely based on safety outcomes, but nonetheless is relevant also in many aspects for effectiveness. But this is a particular example where they used negative controls. That is, they got a large number of drugs for which there's nothing in the label for a specific event, nothing in the literature for a specific event, and then ran them through many, many databases, many, many methods, and just saw what are the operating characteristics of our work. And, you know, the answers were not good. For instance, here's an example of 65 so-called negative controls. We would expect the confidence intervals, 62 of them, to cover 1, and they don't. You know, it's more like 45%. So, you know, the question is, if these are the operating characteristics of the work that we do, how can you make reliable, credible decisions, you know, based on this work? And if you look closely at the regulators' the publications they put out, they have that concern. And you know, I put their words here, but my words on that are, you know, we need to be convinced that the operating characteristics of this research is sound, so that when we run a study, how do we know that it's reliable? You know, when we have randomized RCT and we start with a certain size of type 1 error, we understand our risk of making a false claim. But how do we do that with observational research being the core question here? Now, there's plenty of research in the literature, papers we get that just say, hey, observational research gives you wrong answers, it's bad. Here's one example from Hempkins looking at mortality, and they pulled a bunch of studies for which there were RCTs and real-world studies, and then they lined them up, and that circle at the bottom right says, well, the observational studies overestimate relative to the RCTs. However, every time we see one of these, we often see someone come in and say, no, if you do the analysis right and do more of a targeted trial approach and other things, the observational studies often give you the same answer as the real-world studies. And this is a reanalysis by Jessica Franklin at Harvard. So I think the jury is still out. There's definitely a lack of understanding of what how good, reliable the RWE that we are doing is, and that's a critical question we need to reassess. But I also want to talk a little bit about improving our best practices, you know, as statisticians, and some growing opportunities we have with real-world research. So that's where I'm shifting to over the next slides. So this is a study to sort of reestablish where we're at today that I really am happy to see going on. This is funded by the FDA. It's a trial replication project that Harvard will be doing to take RCTs. Some are ongoing, but they can then do the real-world data without knowing even the results of the RCT. And the kinds of questions they're trying to get at are, you know, when can one study drug effects without randomization? Are there specific disease states, outcomes that are more reliable and valid? How? Is some methodology better than others? And really understand why some studies fail to replicate and why some do replicate. And this, the idea here is to get everyone comfortable moving forward about the best way to use RWE when you want to make reliable decisions. So my question is, how do we get better in this area? Dr. Bazou talked about designs and disruptive designs, and I think that's a great proposal we have there. I'm going to only talk about designs and data briefly, but I do mention them here because they are critical, maybe even more than analytics, I don't know. But my interest in mostly today is going to be on the analytics side, and mentioning only those two briefly. But just want to acknowledge that these are all key pieces if we really want to have reliable decision-making, regulatory grade from real-world data. So pragmatic design, somewhat like was mentioned earlier, have been proposed as a bridge between the RCTs and the observational world, because you have the randomization up front, and then you let it be as reliable or as real world as possible moving forward. And you know, maybe that evidence bar needs to include such randomized trials. And I largely agree with that statement. You know, does it need to go further? Well, I think we have some work to do there. My point on all of these longitudinal studies is that there's a lot of work as statisticians that we can get better on here. So this is an example we published a while back from a pragmatic study. It's from schizophrenia. The y-axis here is reduction in symptoms, schizophrenia symptoms. And this is comparison of atypical antipsychotics to typical antipsychotics. And I just picked up the data and ran 3 common analyses to it. An intent to treat, which found a p-value near 1. On-drug subset that's throwing data out after a switch, you get a p-value near 0. Or if you just look at the complete errors, you get a p-value near 0.5. You know, and the problem is not the design, the problem is not the analysis, but the problem is, you know, just emphasizes we need to have clarity in what our estimates are before we start this. That is the population and how we're handling the intercurrent events, such as medication switching. And some of these questions, if you want to handle post-randomization switches in your analysis, are going to require more complex methods like marginal structural modeling, G-estimation approaches that handle time-varying confounding. And I think these methods are not utilized and understood as well as much as they should be, in part because we never do step 1, that is really understand what we are targeting with our, you know, setting a clear estimate. We typically start with, well, I want to do a propensity score matching analysis, intent to treat. You know, that may be correct, and it may not be the right thing to do in that situation. So methods matter. I'm not going to talk about data because I really want to focus on the analytics, but just to say quality of the data is critical, and we have issues with quality of claims in EMR, and at the same time, we're now trying to investigate what we call non-traditional data, which is where the field is going. So that's a whole other area, not my area, but a critical area we need to address. Moving back to the analytics in for the rest of my talk, again, there are many statistical challenges, real-world data. Number 1 issue is, of course, confounding with the lack of randomization. We have bias. If we want to do really causal inference, we have assumptions that we must make. One is no unmeasured confounders. One is we have positivity or, you know, sufficient overlap. And of course, we use the correct models. This is just a typical example I like to show to get people comfortable with what real-world data bias looks like. So this is bleeding rates in PCI patients undergoing stents. And this is the rates of bleeding we see, and we have a large RCT on the left-hand side, and we can see our treatment had higher rates of bleeding, and so this you know, goes into the label as a risk for this particular treatment. A couple of years later, we go out and look at the real-world data, and what do we see? We're going to see the exact opposite. We're going to see a protective effect of our drug out on the market, which we know is not real, or we believe is not real. We believe it's due to bias because the physicians know those at risk of bleeding are going to be funneled away from the new treatment. So typically, how do we handle this? To date, it's with the methods such as the propensity scoring. But the question then becomes, what's the best method? Lots of people just say propensity score matching is the way to go. But again, stratification may allow for broader population. You don't throw out as many patients. Perhaps regression, it's easier. You got to think about the calipers. We can do inverse propensity weighting. We can match directly using the Howell-Novus distance in X space, high-dimensional propensity scoring, and there's greedy, exact matching, IVs, prognostic matching, with replacement, without, optimal matching, doubly robust methods. Lots of options here. The bottom line is we don't know which is the best method to use for comparative effectiveness. There's lots out there, so we started to address that. You know, are we even doing the right approach for the basic bias adjustment method? and did some very extensive simulations that Tony Zagar published of the first piece of last year. And the bottom line is there is no gold standard best method. The best method depends on what the data looks like, the data scenario. So, you know, we're starting to think, well, maybe we need to borrow ideas from other areas of statistics, like predictive modeling, where you have, you know, cross-validation and model averaging, and those techniques might help us in our real-world data field to do better. Because if there's no single best method in every scenario, let's let the data drive. So this is the type of thinking I'm thinking of. You know, how can we do better from an analytics perspective? And this is just a slide to summarize that model averaging approach. And this is some of our simulations showing that, yes, indeed, using a frequentist model average, we seem to get the right answer a little bit more often than we do if you pick your single best method and just run with it despite what the data says. Another huge area, in my opinion, which is under addressed is the unmeasured confounding that I think statistics needs to handle better. Because this is something we always have to make an assumption about. What should I do about the unmeasured confounding? And the typical answer I got and gave for years is, well, just mention it as a limitation in the discussion section of your paper and move on, because guess what? That will get it through and that will get it published, and I've done it and it works, okay? However, given the criticality of this assumption on all the inferences we're making, I think we need to do a better job. And over the last 10 years, there has been a great increase in the number of potential methods that we have to do sensitivity due to unmeasured confounding. So I just want to take a minute and share some of this, the concepts here. You know, everything depends upon what you know about the unmeasured confounder. You may know nothing at all. You don't even know what the unmeasured confounder is or if it even is there. Okay, well, there are some methods that even apply then, such as IVs or instrumental variables. There may be in a lucky case of the internal where, you know what, you do know what that major unmeasured confounders like to be, and in fact, you can go get it from a subset of patients in your data set. Well, that opens a different set of methods that are very useful, such as the Bayesian analysis I'll show quickly. The growing area here is on this external. There's tons and tons of data we have out there in the literature. Can we incorporate that into our analysis to adjust for unmeasured confounding and do a sensitivity analysis? And one of my colleagues, Sean Zhang, has done this through the Bayesian modeling. So as an example on this next slide, you know, this is an example of how Bayesian twin regression modeling incorporates extra information from data that you don't have in your trial. You may have your standard model based on the claims data that we have, where you're fitting based on your cohorts and your covariates you have measured, and then an unmeasured confounder that you don't have. You might be able to use prior information from the literature, or you can build in the second model, which has your unmeasured confounder here. In this, our example, there was bone mineral density and osteoporosis, where we have that actually in public databases or in other observational studies. We actually can build our model using the same set of covariates and in a Bayesian manner incorporate the two. So we can incorporate the information from the other studies into our analysis as sensitivity. So a lot of innovation here going on that I think would help us move forward and create more credibility. Last thing I'll mention is the growing area everyone hears about is machine learning, predictive algorithms for precision medicine, more individualized medicine. And I do think this is a great area of growth, and that we need to have clear best practices in, because this is a question everyone wants to know. You know, for this patient, what's the best next step on the treatment pattern for them? Can I use other real-world data, since it's so we have hundreds and thousands of patients' data? Can I use that to help me make a better decision on the treatment path? And you know, what we're seeing is that yes, indeed, we can. You know, that if, you know, here's an example that we ran where the actual prescriptions are on the left, you know, which treatment groups, treatment classes are being used, and if we used our predictive, you know, algorithms to optimize treatment outcome, what would the treatment patterns look like? And this is an example in diabetes, and they would be different if we were really out to optimize treatment. And we would be able to get an increase in response rates if we optimize treatment. So it's telling me there's opportunities here through real-world data and statistical algorithms to really improve patient outcomes. So then let me just close by saying, I think we do need to improve the foundation, the operating characteristics. I think that's the critical thing for establishing the credibility of real-world evidence. We haven't done a good job of that to date, but I like where we're headed with many things that are going on to date. As statisticians, there's lots of opportunities for innovations. Bias control is one I mentioned. Unmeasured confounders another, and precision measurements another. These are three areas that certainly the pharma industry is interested in understanding and doing better in. So with that, I will hand it back to you, Dan, and look forward to the discussion.
Great, thank you. That was a great presentation of where we are, some of the most important issues, and how we should start to address those issues. So with that, I'd like to reintroduce Kelly Zhou, Senior Director at Pfizer and ASA HPSS Chair, and let her give some of her comments and start the discussion. Kelly?
Thank you. Thanks, everybody. First of all, if you would like to ask questions through the Q&A box online, please do. I've already collected several comments, and we'll get to them and bring to the presenters. In addition, we do have a large number of attendees, actually right now at 313, so thank you all for attending. So based on these two presentations, as a discussant, I think I have a few thoughts, and in addition, earlier this week, there was the Duke Margolis workshop in terms of real-world evidence for regulatory framework, so hopefully many of you have already attended. So combining all the interests and the mandate under the 21st Century Cures Act, I think both presenters, the contents are extremely timely. So let's first talk about Anubhav's presentation. So the main keyword I got from the title, of course, the disruption aspect, but the keyword I really think that stands out is the word infrastructure. And why is this so important? Because drug development is a very well-regulated process, and from end to end, as Anubhav said, it's a phase approach and hypothesis testing driven. And however, given that the real-world data are the type of data that are outside the RCT, he has proposed a really interesting and hopefully workable alternative. And one of the questions online was, what is OIR? So Anubhav, you gave us the abbreviation, and you explained that in your Health Affairs article. So if you look up his name and the Health Affairs article, you will know that OIR stands for Only in Research. So this is the alternative infrastructure and that he has proposed. And there are a couple of questions regarding that process, so we'll get to that later. However, this is quite thought-provoking and groundbreaking, and hopefully this will be a potential alternative given that the drug development process has been so well defined from early phase to late phase. So given that, I'm going to move on to some comments on Doug's presentation. So what's really reassuring is that Doug has emphasized so much about quantitative skills, and particularly the type of analyses that are common, or some may or may not be that common. So he has given us a sample of all these potential tools that we can leverage in the context of leveraging real-world evidence. Now, back to Duke Margolis' presentation workshop earlier this week. Interestingly, when we think about real-world data studies, according to one panelist there, it's not just the traditional observational studies that we are familiar with and saying that we just leverage existing type of data. Nowadays, such kind of studies can be quite complex. So real-world data studies could be observational, could also be pragmatic trials, as well as registries. So Doug has talked about quite a bit about, reducing or minimizing or eliminating biases, and he has also talked about pragmatic trials. The word that stands out from his talk, from the title, is really innovation. However, for innovation, the other side is how well, the new innovation agrees with what RCT can provide. So he has talked a lot about reliability and reproducibility, and about replicating, results based on RCTs using additional information or alternative information based on real-world data. So the final takeaway that I have is that, there could be infrastructural disruptions, but at the same time, quantitative skills and, statistical thinking are still very important regardless. So hopefully the regulators will balance all these, considerations and develop an infrastructure that can really encourage and, provide the opportunity for academia, industry, government, all sides to leverage, all the skill sets for drug approval process. So that's pretty much my comments based on their presentations. Now I'm going to get to the questions. So we have about 20 minutes from now until the end of this presentation workshop. So if you still have questions, provide them online through the Q&A box. So I have a first question for Anirban. The question is back to your health affairs article that was so well received and so well publicized in the media. If you read that article, the author, Anirban, wrote the following. He said that to curate real-world evidence to meet the quality standard of randomized controlled trials, and to do it in a timely fashion so as not to delay access to valuable innovations, requires disruptive innovation, as he said in today's presentation. So given that the RCT has been considered the gold standard in terms of evidence, in the new paradigm that you have proposed, which is the OIR infrastructure, the real-world data studies for regulatory purposes may require innovations such as data capturing systems, fit-for-purpose data sets, and data science talents. So the question is, how can statisticians and data scientists collaborate closely with interdisciplinary team members as part of your proposed disruptive innovation paradigm? Anirban?
Yeah, thanks, Kelly. Good question. I think absolutely. I mean, such an infrastructure needs a diverse talent of people working together, not just clinical trialists, but statisticians, specialized in other aspects of design and analysis, like predictive modeling and instrumental variables. You need epidemiologists, economists, decision scientists. I think there are a lot of methodologists that work that come from different background and bring different ideas to the table, and we need kind of these teams to work together. My opinion, in industry, we see a milieu of such teams already, and especially that the real-world evidence is getting more popular. At least people are starting to thinking of how to incorporate them in decision-making. I think, especially in pharma, we see these teams are being formed. You know, in our Choice Institute, our PhD program are really an interdisciplinary program where they learn economics and biostatistics and quantitative decision analysis and all those things. But I think from the government side, from the regulatory side, I think we would love to see also more diverse teams working towards this problem of, you know, producing evidence that would be then used to inform so many different type of decision makers in the system that rely on that evidence to make, you know, very important decisions that translates to patients value.
Thank you, Anirban. Kelly, are you still there?
I can go over the questions, and Doug can, you know, chime in at any point, and if that's
yeah, that sounds like a good way forward. Why don't you do that?
Sure.
Yeah.
Right. Anirban, I have a question for you as well. You know, just coming from the pharma side here, you know, I love the proposal here. One of my questions has to do with the economics of such trials in what I would call economic equipoise for that trial that you would run. You know, the issue becomes in these pragmatic studies, who pays for the drug? Because you have to have, you have to decide whether you want to create that to be a balanced cost issue or not. You know, when you do that, you're influencing. It's not 100% real world. You have to make that choice, and so that's what we wrestle a lot in the pharma. We feel if it's randomized, we have to pay, but if we have to pay, we can't let our drug be free and the competitor not be free. So how do you think through those issues?
That's a good question. The way I envision is that this is a market access. So you are allowed to market your drug in the OIR phase with the OIR designation, but in a random subset of patients. And so the payment for that drug would be made by, you know, insurance companies, payers, and patients. But again, as I briefly glossed over the fact that there are a lot of economic forces and incentive built into the system where you wouldn't see the prices of this new experimental drug at the same point that you see in a fully approved marketed product. So there is something in between where there is a tremendous potential for this drug to come out and help patients, and but at a price that is obviously more than the marginal cost of producing the drug from the company's perspective. Otherwise, there is no point for them to market the drug, but way below what would happen if they were fully marketed. And so certainly we don't want, you know, with a population access that the manufacturer should be, you know, putting the bill forward for all those use. It has to be, you know, a system level market access issue, and this is, you know, the healthcare plans and insurance companies should be paying for it. But again, you know, because of the randomized access, the use of the drug in the real world setting would open up the opportunity to do all sorts of you know, real-world evidence with the randomization at the backing. Now, just to add to that, I mean, I think there's a lot of questions about, you know, how would we calculate sample size, or how would we enforce exclusion-inclusion criteria in these kind of studies. I think all of those are exactly the way you would do in a big pragmatic trial, or in any kind of research scenario. All this infrastructure does, it gives randomized access. So some people are using it, or have the potential to use it, and some people do not. And when you set up a research question, you can take the population and apply your exclusion inclusion criteria, just like prospective observational study. You can calculate sample size, like how many people you need to study really to answer a much more nuanced effectiveness question, and do that study. So nothing of those designs changes. It just, we take out this solicitation-based artificial randomization and make the randomization to be a population level one.
Thank you. Thank you, Anupam. Any other thoughts or questions? Otherwise, I'm going to ask Annabelle one more question because we still have some time. So the last question is, Annabelle, you wrote in your article, the key challenge lies in curating real-world evidence to ensure the right regulatory decisions can be made with that evidence. So you brought up a good point, and then also this point was so emphasized during the Duke Margolis workshop, is about the curation. So we know that traditional statistical quantitative training, we learn many methods for analyzing data, but the curation aspect may or may not be that strengthened. So how much effort there is for this aspect, and what do you think would be the necessary steps and skill sets for curation?
I think, you know, the way I think I was talking about curation is the idea that, you know, often when we do real-world studies, we rely on claims data. Obviously, we cannot rely on just claims data to answer, you know, real effectiveness questions. And the question is, can we develop, whether it's IT infrastructure, EMR-based, or in prospective observational studies, to actually kind of, you know, design questionnaires for a certain number of patients to answer, you know, relevant outcomes or measure certain outcomes, and reduce the measurement error in those. I mean, and to me, the curation of the data can mean many things to many people. To me, it is really about a reduction of measurement error. That what we are studying, what we are seeing is, I can rely on it, and I think, you know, there is a lot to be desired in that front in terms of the quality of the data, at least in this country, how we could kind of link data sets across different settings for the same patient to really see the effects. For example, you know, a schizophrenia drug coming out has huge implications for their cognitive abilities, but then, you know, unless we really go and have spent a lot of money understanding how that affects the criminal justice system or some of their recidivism behavior, we do not have that data. And so linking across databases, you know, improving the quality of those linking is I think fundamental to do you know, good real world data evidence, or generate good real world evidence.
Thanks, Annabelle. Very quickly, I already said that I was going to ask the last question, but there's such a great question coming up that if you can just say very briefly respond to this question. So the question is, as a trial statistician, we're worried about the moderate efficacy signal being drowned out by the noise. There's a concern for pragmatic trials. Wouldn't this be an even bigger concern for your OIR approach?
So that's a very good question, and I know you have to think about the reason it gets drummed out is because you think there is heterogeneity. And if you really are concerned about heterogeneity, then you should be actually doing more heterogeneous hypothesis testing, which would obviously require much larger sample size, and which just cannot be done in a small clinical trial-based approach. My take to this is that, you know, the whole idea of so the access question is always a population level question. You know, should I give access or not? The clinical decision-making question is often an individual level decision-making question. Should this patient get this drug or not? And these are fundamentally different questions. And you can think about, we need completely different methods and data to analyze for these questions. But I think the OIR infrastructure sets up well to provide that data to answer these different disparate questions. So, from an effectiveness trial, yes, I mean, one of the things that I always worry about, that if you are doing a pragmatic trial and really incorporating a lot of heterogeneous subgroups and randomizing them, then what's the point in looking at an average effect? It certainly doesn't help or inform clinical decision making, where the doctors have to really make a decision about individual patients. But it can certainly help in answering the question, what is the value of giving access to a drug, which is a population level question. But it can set up to kind of, whether through predictive modeling or through other sources, to really get to the nuanced, algorithm based or kind of risk equation based information to get to that nuanced subgroups to make individualized decisions. So I think the key thing about this infrastructure is that, does it have the necessary parts to exploit to generate the data needed for very different decision making?
Thank you. Thank you, Anubhav. So I would suggest everybody, take a close look at his proposed infrastructure in his article, and potentially having a journal club. And of course, if you have lingering questions, perhaps you can also contact Anubhav for future discussions in terms of alternative framework. In the interest of time, I must move on to Doug. Doug, there are a few questions online, so I'm going to just combine a few. First is really about real-world evidence associated with, literature and publication. Even though, you mentioned about several different types of real-world data studies, such as observational as well as pragmatic trials, there are a couple of questions. So the first one is, the Open Science Collaboration recently replicated 100 psychology studies, and unfortunately, only about 30% of such studies had the same outcomes, mostly due to publication bias. We know the fire drawer syndrome. That's my comment. So do you think that publication bias in the medical literature is about the same, or it's even worse than that? And in addition, if you consider deriving real-world evidence by examining the literature, how would you recommend adjusting for the bias? I know that you already talked about Bayesian framework, Bayesian methods and others, and bias reduction methods. So for audience who may or may not be statisticians, can you say a few words about the relationship between real-world evidence, derived from real-world data versus, what's out there in the literature?
All right, there's a lot in that question, but I will do my best here. So if we're talking about randomized trials, I don't think there is currently today a big issue with file drawer problem, because we register, at least if it's sponsored by Pharma, we're going to register our clinical trials at ct.gov, and it'll be out there before we get the results. However, that does not happen with observational research. That's one of the issues in the 2017 guidance that the field is pushing for. Hey, if we want the same reliability, credibility as RCTs, let's handle that in the same way. If you're going to be making comparative statements and claims about your observational research, then state it prospectively and put it out there so that we get away from the issue. Well, we just publish when we get the positive results. I know each individual company has addressed this. I don't know across the industry, but I do know that, we have our own principles of medical research where we try to say, what, we are going to publish if it's about a drug on the market, we have an obligation to publish it. So we will do that, but that is an issue I think that they're trying to address with the 2017 guidance.
Thank you, Doug.
Oh, yeah, what was the second part, or did I fully address the question there?
So if you derive real-world evidence, if you examine the literature, and also if you have real-world evidence, how do you kind of adjust for bias potentially one way or the other?
Well, I guess this goes back. I'm going to answer a related question, which I think gets at this issue, and that is, how do we get comfortable with the real-world evidence? And so in this question, it's putting it next up to the literature. In my example, it's putting it next to the RCTs results. And I think when you talk about any kind of change, the natural question people are going to have is, well, how would that differ from what I'm doing now? So in our case, how would the real-world evidence differ from what I'm getting with the RCT evidence or that's out in the literature? And so I think we need to have two things on this. One, we need to provide an answer for that, and that's why the things like this replication project I mentioned are critical, because it'll help people answer that. When do you get the same result, and when do you not get the same results, and why? So I think that's important. And then secondly we typically don't design our real-world evidence to try to come up with the same answer, because we have different goals. So it's okay when they don't match, because they shouldn't always match the way we're designing our RCTs. That people need to understand the objectives, the estimates, and everything, and of the observational research, and it may differ from what's out in the literature study or in the RCT. And so that needs to be taken into account when people just post, well, we did get agreement or we didn't get agreement. There has to be a systematic alignment of objectives and estimates and the like, the targeted trial approach, if you really want to compare the two side by side. Otherwise, I don't think you can do it.
Thank you, Doug. We only have 1 minute left, so in the interest of time, thanks everybody for joining, but I'm going to return it back to Dan.
Thank you, Kelly. Again, I want to thank our panelists for giving two great presentations and having a really excellent discussion on some of the most important topics and issues in this area. I'd also like to thank Christy Trang-Stein for helping organize, and Liang Zhao Zhu, who also had the idea for this this particular meetup, and Kara Hackman, who helps with on the technical side. Part of the reason for the topic of this meetup was based on the survey that we do at the end, which is only 3 questions. So I do encourage folks to answer those questions, in particular if you have topics you'd like to see in the future. And with that, I'd like to thank all the attendees, because obviously the most important part is to try to get important and useful information to you. So with that, I'd like to wrap up, and again thank the panelists for such a great presentation, and thank everybody for attending, and we'll hope you tune in next time. Thanks again. Bye-bye.
Thank you. Bye.
Thank you.